Probably best in your notes if we go back to this slide. So we were talking about protein turnover and the amount of protein in the body. And last lecture I mentioned that there's thousands of different proteins in the body. And as you can imagine, some of them are a lot more prevalent than others. So in your notes, I want you to write down these 4 proteins. These are the most common proteins in the body. So almost half of the body's protein is contained in just 4 different proteins. Almost half of the body's protein is contained in 4 different proteins. Slide 8. One of them, can you guess any of them? Most common proteins in the body. Proteins. Collagen. Most common protein in the body, collagen. What does collagen do? Hold cells together. We all need to thank collagen because we're multicellular organisms. No collagen, we'd be yeast. Okay? A karyotic single-cell organism. Right? 2 other ones, actin and myosin. Last one, hemoglobin. Extremely common. So one of the points of that is, wow, if you're breaking down proteins, kind of likely it's actin or myosin, right? Very common. And the reason not as much collagen sometimes, it's extracellular. And in 329, hopefully you'll talk a lot about collagen because its metabolism is dependent on things like vitamin C and iron and some of the other nutrients to do that. Sorry about that, I missed that. Didn't miss anything here. All right, so let's move on. So now let's talk about the catabolism of the amino acids. We talked about their interconversions, how they make other nitrogen-containing molecules. Here, let's talk about catabolism. 57% of amino acids absorbed by the liver are catabolized, broken down like this. The liver is the main site for amino acid catabolism in general, but it's not the major site for breaking down branched-chain amino acids. So we'll talk about why they're unique in a few minutes. Now, during this process of catabolism, one of the things that happens is you end up with nitrogen. You end up with amine groups that can become ammonia. So the liver has a way of breaking down those amino acids in a safe way. And that's called the urea cycle. So we'll talk about that in a second. So what happens in nitrogen? We'll talk about urea cycle. One of the things though that happens is the nitrogen, the amino acids are either, they can go through transamination reactions and deamination reactions. We'll look at that really quick. And then we'll look at what happens to the carbon skeleton. So one of the things to keep in mind is, like, we can go alanine to pyruvate, back and forth. We can go, matter of fact, aspartic acid becomes oxaloacetate, and vice versa. And glutamate, alpha-ketoglutarate, I wrote that before. And if you just look at that, would you guess any of these are essential amino acids? Essential amino acids. No, they're relatively easily made. So these are non-essential amino acids. But one of the things to think about is, well, when you're switching, when you're going from a molecule of like glutamate, and you're making an alpha-ketoglutarate, what happens to the nitrogen? Because if you take this amine group off, right, it's basically ammonia once it becomes free. So our liver minimizes that. And one of the ways it minimizes that is it pulls this off but adds it to another molecule. Doesn't let it go free. So it's transferring it, or it's transamination reaction. And that's what this is showing. This is a transferase or transamination reaction. So look what's happening. This enzyme, alanine aminotransferase, holds on to glutamate. Let's say this is glutamate. I have some great models here. I think this might work. Yeah. Here's glutamate. Okay? So this enzyme is obviously alanine aminotransferase is my hand. Holds on to glutamate, pulls this part off. This alpha-ketoglutarate. Releases this. Now, it hasn't let this go, so what does it do? It adds it to pyruvate. It forms alanine. So the amine group's never released. It's transferred. That decreases ammonia production. So there's some really important transamination reactions, especially the one done by alanine aminotransferase. It's called alanine because that's a common amino acid. But look at this, shows it going forward, but it'll also go backwards. Again, alanine, glutamate, not essential because we can easily make them during these transamination reactions. In general, the essential amino acids, not a part of transamination reactions. Yes?
Does the same thing happen with, like with pyruvic acid, how it has 2 double bond oxygens? Does the same thing happen by bringing an oxygen down to the alpha ketoglutarate acid?
It's the same thing.
Since it goes from 2 to 3 on the bottom and it goes from 2 to 1 on the top.
Does it say, I think, you mean does pyruvate become alpha ketoglutarate?
No, because the pyruvic acid has 2 double bonded oxygens.
Right.
And once it's transferred to alanine, it only has 1. And on the bottom reaction, it's opposite. It goes from 2 double bonded oxygens to 3. So does the same kind of process happen with the oxygens?
Yep. Yeah. Good point. Yep. Yeah. So this is a liver enzyme. Remember, I said some enzymes go up in the blood if the liver cells are breaking down. This is one of them. So when you're looking at clinical lab values, sometimes you'll look at ALT levels, and you'll look at AST levels. You'll look at some of these to say, oh, that's normal. If it's abnormal, you're like, oh, something's going on in the liver. Okay. Here's the other kind. This is a deamination reaction. As the name implies, the amine group is just removed. Here's threonine, for example. The amine group comes off. It becomes ammonia, which is basically NH3. Now it's basically free in the cell. Ammonia and ammonium ion go back and forth. So this is actually ammonium ion, because now that nitrogen has a 4th bond. That gives it a formal charge of a plus, right? But they go back and forth. So ammonium ion, ammonia, not helpful. Okay? So accumulation. So the liver gets rid of ammonia by making the urea cycle. So the trick is the liver can do this and then take that ammonia, use it to make urea. So here's the urea cycle. And this is how a lot of animals like us and other similar animals deal with their nitrogen. Some other species don't have urea cycle. And that makes their nitrogen metabolism completely different from ours, but I won't go into all the comparative physiology. But here's the urea cycle. So a few things you're going to need to remember. Urea cycle in the liver. And here's the first, and then it makes urea, puts that into the blood, and then the kidneys can excrete it. So made in the liver, excreted through the kidneys. So urine has urea. Here's how it starts. Here's our ammonia. First step is this enzyme, carbamyl, I don't remember the enzyme name. Ammonia combines with carbon dioxide to make carbamyl phosphate. All right, so all it's done is it's now combined ammonia with carbon dioxide. All right, so we've essentially taking away the ammonia and then puts that into the urea cycle. So here's the first part of the urea cycle. Bear with me. Don't have to remember this, just parts of it. It'll continue to cycle even if we don't know what's happening, right? So here's the first part. Here's carbamyl phosphate. First step combines with ornithine, right? And so what's happening if we follow that nitrogen group that's in blue, right? Now it's part of citrulline. Citrulline combines with aspartic acid. Here comes another nitrogen. So think of the point of it is to take nitrogen, put it into urea. That's what the liver's doing. Now we have a nitrogen here and a nitrogen here. Arginine succinate becomes fumarate and fumarate and arginine. Right? No nitrogens here, so the nitrogens are retained. Now look what happens. Arginine becomes ornithine, and as that happens, urea comes off. That's one cycle. So one urea cycle generates urea that has two nitrogens. And now the urea is secreted from the liver, goes into the blood, and the kidneys can excrete it. Did it take energy? Yeah, how many ATP? 2 here. 2 here. 2 more. 4. Takes 4 ATP to make a urea molecule. You ever heard some people say, oh, it takes more energy to process dietary protein? Is that right? Here's one reason. Yep. Increasing dietary protein can help, in some people, maintain ideal body weight. It's more energy dependent than especially lipids and also carbohydrate. So here's one reason. Now, this is if everything's working right. If the liver becomes dysfunctional, ammonia can accumulate, and that's a problem. Have you ever heard of a condition? If you haven't yet, you will. Hepatic encephalopathy. That's when basically the brain and central nervous system are being damaged by ammonia. If the liver becomes dysfunctional, got to watch protein intake. You tend to minimize protein intake, and that can decrease ammonia accumulation. You'll talk about that in clinical. Have you ever heard this? If you eat too much protein, it damages the liver and the kidneys. Probably not true, unless it's really high. So in general, a healthy liver and kidneys can handle significant amounts of dietary protein. I'd say in general, ballpark number, a lot of nutrition people that have a background in nutrition and nitrogen metabolism will say about 300 grams of dietary protein. That's probably a ceiling. And that's when it can be problematic. And I would hope so. That's a lot of protein, right? Right? Very, very, very few people would need that. Okay? Your little brother doesn't need that much. Okay? That's one of the most common questions you get, right? Oh, you're in sports nutrition? Yeah. What kind of protein should I take? Salmon. What? So very common for people to eat a lot more protein than they need. So we'll talk about requirements in a second. Here's a point, and then we're gonna switch. We're gonna move to the next lecture. If you take a urine sample and you're looking for nitrogen, and this is the main way we get rid of nitrogen is in the urine. That's why we drink so much water. That's why another reason water is so important for us. That's how we purge nitrogen. Some animals don't need much water. They don't do the urea cycle. Birds, for example, This is fascinating, right? No, urea cycle, they get rid of their nitrogen as uric acid. They don't need water to do that, so they don't need as much water as we do. But here's the point of it. Here is major forms of nitrogen excreted in the urine. This is from another text. This isn't in your text, but this is a good one. Look at the different forms we have. Urea, ammonia. This is not right. This is ammonium ion, right? We all know that. Creatinine, that's from creatine. And uric acid. What's this famous for? The association between elevated uric acid and gout. All right, they use gout, rich man's disease. It's not. Then they figured out it's not probably from dietary protein. It's from some other things in the diet, but gout is accumulation of uric acid and an immune response that causes intense pain. So it is related to nitrogen metabolism. But the one major point here is major form of nitrogen in the urine is urea. About 85% of urinary nitrogen is urea. See why that's so important, the urea cycle and the liver's functioning normally? And then you have some ammonia ends up in the urine and some creatinine. And the creatinine comes from creatine in the muscle. So creatine goes to creatinine and then it goes into the blood. So if I ask you, how do we get rid of creatine? We make creatinine and put it in the urine. You'll see that on some lab values too in clinical nutrition. Sometimes you use creatinine to look at muscle wasting. It's a slow process, but if you see enhanced creatinine, it can mean muscle wasting. And then uric acid we talked about, but the main point is urea. Okay, I'm going to skip this one. You like that? Let's talk about what happens to this. So this is the remaining skeleton. So, right, we took this off, did urea cycle with it. Now we got these carbons. That's called the carbon skeleton. What happens to them? Well, it depends. This verbiage explains this. And I'll put this in the study guides that I'll post tonight after my salmon. If you look at this, it's showing this a single amino acid becoming an amine group and carbon skeleton. The amine group. So you want to be able to give me this and tell me the specifics of what's going on here. The amine group can become urea or can stay as ammonia excreted in the urine. But some of those amine groups can be used to make non-essential amino acids. Don't say essential. Non-essential amino acids. So basically this could go here or it could do this. All right, that's what the liver can do. Now what about this? It can be used for energy. It can be used to make glucose or ketones. It can be used to make fat. I'm going to ask you under what conditions what happens. So in other words, when diets are high in energy, protein and carbohydrate, some of the excess amino acids are used to make fatty acids. So if a person's consuming high calories, including too much protein, some of those amino acids are used to make fat. And that's in the liver. So the liver's making the fat, and then it'll be transported to fat tissue by VLDL. So if I ask you what happens during fasting or starvation, don't say that. Right? All right? And this is happening during high calories, including high protein. The other ones, low energy, low carbohydrate intake. When diets are inadequate in energy or carbohydrates, those carbon skeletons can be used for things like alpha-ketoglutarate to support the Krebs cycle. Right? So look at this as, why does this say CO2? That's Krebs cycle. Remember those CO2s that come off? and then we use oxygen, that's called respiration or breathing. Right? That's Krebs cycle. And then this is gluconeogenesis, ketogenesis. Starving or lack of carbohydrate, for example. A lot of those amino acids can be used. Energy production, glucose, ketones. Now in that case, some of those amino acids came from skeletal muscle. And the liver's converting them and using them. Let's talk about branch chains really quick. Years ago, they did this experiment where they said, let's feed these lab animals, these rats, a mixed diet. And they said, let's take a blood sample going from the gut to the liver, in other words, portal vein, and they gave them a mixed diet that contained approximately 20% of the protein was branch chains. That's a pretty typical amount. And then they thought, well, 20% going in, portional coming out. That's not what happened. They went, wow. Of those amino acids leaving the liver, 70% branch chains. So that told those investigators the branch chains not really metabolized in the liver. They're used for protein synthesis in the liver, but they're not metabolized in the liver. They're on their way to muscle. The muscle metabolizes the branch chain amino acids, unlike the liver. And this is what the muscle can do with them. There's the branch chains, isoleucine, leucine, and valine. If you want more details, look in your text. And there's a lot of steps here. The points to this are, this is the skeletal muscle using branch chains for energy during aerobic conditions. See what they're metabolized to? Succinyl-CoA, acetyl-CoA, molecules that can be used for Krebs. Okay. The reason this can happen is, see that transaminase right there? That is called a branch chain amino acid transaminase. It's high in muscle, very low in the liver. So skeletal muscle can take that amine group off of the branch chains. And then you end up with a branch chain keto acid, and it's metabolized. Got it? That's why sometimes you'll see people say, oh, I take a lot of branch chains for skeletal muscle, whatever. And that's what they're driving at. That's why they're a little bit unique.
What's the BCK80?
Branch chain keto acid dehydrogenase. You don't need to know that.
Oh.
That's 329 stuff. I typically show this in 329. I talk about the vitamin dependency of those enzymes and a thing called muscle syrup urine disease. Good there? Let's go to the second one. Protein lecture 2. Oh, yeah. The review sessions. So I'll give you study guides tonight and tomorrow, Friday, Saturday, Sunday. I'm thinking let's do a couple of them Monday. We do 2 different times on Monday. I don't think I'll be able to get pretty much all of you, I think. So let's do Monday at 10 a.m. and Monday at 2 p.m. 10 a.m. and 2 p.m. So it's kind of an office hour review session, and I'll find a location. So if you're not sure, just come to my office. If you can't make those times, let me know, and I'll try to do something else. But that way you've got still at least 24 hours to go back through stuff if you need to. Okay? I'm not even going to look at my schedule because I'll modify my schedule to make that work. Let's talk about this one. Let's talk about what proteins we need. And I can go through this in a way that hopefully will work. Here's one of the questions. How much protein do we need to eat each day? Right, a lot of times it's, the number. Well, what's that based on? That's based on, in adults, we need to consume adequate protein to replace losses. That's basically it. If we're not trying to add muscle or add weight, we just need to replace the protein we lose. So in adults, we need to eat adequate protein to replace losses. So this was a clue. How do we lose protein? Right, we lose it in urea because protein's used to make urea. How else do we lose it? We lose it with things like sloughing of cells, like slough skin. We lose protein when we lose fingernails, toenails, hair, a little bit of protein in sweat. Things like that. Most commonly, though, urea. So in other words, think about it this way. If I stop eating protein, do I still make urea? Yep. Doesn't stop. We're consistently breaking down protein. So we need to be able to replace that protein. So that's basically what that means. And we just looked at something like this. Right? So when we looked at a slide before, we said the major form of nitrogen in the urine is urea. This one basically shows the major route of nitrogen loss, urine versus feces. So now you know the major form of nitrogen in the urine, but now if you compare urine to other routes of loss, urine's most significant. Second, in the feces. And that can be unabsorbed protein, or I'll show you another thing that can end up in there, but that's mostly in the urine. You lose some in the skin and stuff like that, but the point of it is we're consistently losing protein through those routes. This one I'm not going to go into, but one point here is in the urine we tend to lose nitrogen-containing molecules, but not protein. Protein shouldn't be in the urine. If protein's in the urine, good chance kidney dysfunction. There's a lot of things that should be in the urine. Protein is not one of them. You have some amino acids, but most of the amino acids are reabsorbed. But again, you tend to have more urea than anything.
Can you toss out protein?
Yeah, I guess you can. I don't know if I'd look at it that way. I'd probably ask you, whether or not the protein is in the urine. It really shouldn't be. Some creatinine. Remember, that's how we get rid of creatine. So that's normal. And then some amino acids, but not nearly as much as urea. Again, urea cycle is extremely important. So now let's talk about this, because if we look at this and we say, well, how do we know how much protein is kind of going through us in a way? We can measure or estimate how much protein we eat, but we don't really lose protein. We lose nitrogen. So we need to back calculate nitrogen to protein. And you've done this in 210, so this is probably a review. I've got the wrong slide up there, that helps. Protein contains approximately 16% nitrogen. In other words, if I have 100 grams of pure protein here, 100 grams, 16 grams of that, nitrogen. So always remember your nitrogen number is lower than your protein number when you do this calculation. Why is it about? Every amino acid has an amine group. Some amino acids have nitrogen in the side chain. So we say it's about 16%. So you just convert. So if you know the grams of protein, multiply it by 0.16, that tells you how many grams of nitrogen. If you know nitrogen and go into protein, again, your protein number's higher, so you go 6.25 times the grams of nitrogen. This is where we're going. Let's say I take urine samples from people, and I measure how much nitrogen is there. I need to be able to figure out how much protein that came from. So this is one of the key concepts of this lecture. This is nitrogen balance studies. So nitrogen balance studies are a comparison between nitrogen intake and nitrogen loss. Nitrogen balance studies, what are they? They're a comparison between nitrogen intake and nitrogen loss. So the basic definition is like this, based on this equation. A person's nitrogen balance is you're comparing the grams of nitrogen consumed, and that's in protein, compared to how much nitrogen they lost. So you just go intake minus excretion. Have you ever heard this? Positive nitrogen balance. If a person has a positive nitrogen balance, that means they are consuming more than they're losing. And that's what this thing is supposed to show. Nitrogen intake is greater than loss. When would you expect that? During growth, adolescence, through development. You'd want to consume enough protein to support that. So you want to be positive nitrogen balance. During pregnancy, in a recovery stage from an illness or event, you want to have adequate protein. And also those that are training to try to add muscle or lean body mass, they want to be in positive nitrogen balance to support additional lean body tissue. When can you find negative nitrogen balance? In other words, loss is greater than intake. If a person's not consuming adequate amounts of protein, then their losses are greater than their intake. If they're not consuming adequate calories, they're breaking down protein to get energy, and that'll put more nitrogen into the urine and losses, so you'll end up with negative. So that's one of the keys. I'm always amazed. You got to remember, carbohydrate and calories help a person's protein metabolism, so you got to make sure it's balanced. If a person's consuming a deficient amount of essential amino acids, that can compromise protein synthesis and protein retention. That can put them in negative nitrogen balance. So protein quality is important. We'll talk about that in a minute. If there's increased protein losses like from kidney dysfunction, or wasting due to things like bed rest. Remember I said as soon as a person becomes non-ambulatory, non-mobile, their skeletal muscle is going to tend to be broken down, and they'll lose more nitrogen because of that. So where do most people want to be? Nitrogen equilibrium. Eating the amount of dietary protein that they need to replace losses. So in general, that's where adults want to be. So how do you figure this out? You do a nitrogen balance study. And maybe at one point in your life, you'll be one of these, be part of one of these. I personally won't do the research. I'll tell you why. If we look at this, here's just an example of what you do. Now remember, we're going intake minus excretion. How do we do intake? Well, we know how much they eat. So this group, this is 4 individuals, and obviously you'd have a lot more in this study. They ate 12.1 grams of nitrogen. How do we figure out how much protein that is? Times 6.25. So that's about 75 grams, I think. Right? Is that enough? Well, let's see. There's your intake. Here's your excretion. Collect urine constantly. That's mostly urea. But basically, you take the urine samples, you concentrate them, you do a nitrogen quantifying assay, and you come up with this number. The other major route of loss in the feces. So you're collecting fecal samples, concentrating them, typically freezing them, concentrating them, doing nitrogen. You add those up. Here's your intake minus excretion. If you look at the first one, intake minus excretion, positive. Intake's greater. Is 75 grams enough for this person? Yes. They're positive nitrogen balance. Second person, doesn't look like it. Third one, probably. Fourth one, no. You do this in a population, and then you determine under this population, in this population, under these conditions, this is an adequate amount of protein from this protein source. That's how they did the RDA. Lots of nitrogen balance studies in different populations.
Is it bad to have a positive balance if you're not growing, pregnant, recovering, or training?
It depends. It depends on how that puts you on your calorie balance, and then are you getting adequate amounts of carbohydrate and all your other nutrients. But in general, if you're doing all that and energy expenditure is balanced, generally no. Okay? Got that? Right? Let's look at the next one. And this has to do with what I said before. This is obligatory nitrogen loss, and this is the amount of nitrogen lost from the body as a result of the breakdown of the body's own proteins and amino acids. In other words, this consistently happens. Even if I stop eating protein today, I'm going to continue to lose nitrogen. And when that becomes too significant, that's when it becomes lethal, when you can't support nitrogen metabolism anymore. Again, if you're on Survivor, water, alliances, get your protein alliance set up, right? And here's what tends to happen. Initially, what happens is we're losing more nitrogen initially when this is on a nitrogen-free diet, and then we reach kind of a state of equilibrium, right? And so if you continue this, you can't maintain that for a long period of time. But what's happening in this case is your metabolism is adjusting to this. And you're doing things like switching over to ketones to minimize the amount of skeletal muscle that's broken down to get glucose, and that's what minimizes the nitrogen loss. That's one of the things that happens. So, but you're always losing some nitrogen. One of the key points to this is carbohydrate has a protein sparing effect. When they did these studies, this was like done in the 30s and 40s, right? When a lot of the world was at war, and they decided if we have our military personnel in dire situations, what should we put in their survival rations? And they knew they needed to have protein. They tried to figure out how much protein they had to have in there. One of the things they figured out, if they include some carbohydrate, it cuts nitrogen losses in half. Carbohydrate has a protein sparing effect. It can cut nitrogen loss in half if you have at least a minimal amount of carbohydrate. Guess what that amount is approximately? About 125 grams. Right? It helps retain nitrogen. So that's another important part about carbohydrate. I think we do about 2 or 3 more slides and then we're fine. How much protein do people need? Well, a lot of times we say, oh, they need the RDA. Best estimate for the amount of protein required for individuals to maintain protein equilibrium is 0.8 grams dietary protein per kilogram of healthy body weight. Healthy body weight because if a person has a lot of adipose tissue, they don't require as much nitrogen to support adipose tissue as they do muscle. So as long as their body weight is and body composition is approximately kind of in a typical amount, then that's fine. I won't go through this. You guys need to do RDA calculations if I tell you a person's weight, for example. So you can look at this. Again, 56 grams a day it could be considered probably a relatively small amount of protein. Is that enough for everyone? It depends. You got to make sure you're consuming adequate amounts of energy. Otherwise, some of the body's protein is used for energy. So you got to make sure you get enough calories. You got to make sure you get adequate amounts of carbohydrate. And you got to make sure you don't exercise. If you exercise, it changes it. Now this says athletes in the RDA, but there's not an RDA for exercise. So this is basically a comparison. So if you look at this, it says strength training athletes need 1.4 to about 1.8 grams per kilogram body weight. Is that a lot? That's more than twice the RDA. And the reason is because it takes more protein to add and maintain lean body mass than it does fat. So if a person is adding lean body mass or maintaining a significant amount of body mass, they need more protein. How'd they figure that out? Nitrogen balance in athletes. What if you go above that? Probably not helpful. That's your ceiling. So hit that mark and then look at your other nutrients. Endurance athletes, do they need more than the typical RDA? Absolutely. They need more because of changes in body composition and slightly altered energy metabolism. Endurance athletes tend to need more because to support changes in body composition and changes in energy metabolism. In other words, they can metabolize more amino acids for energy than sedentary people. Let me give me 30 seconds. When you talk about protein quality, like if you say, okay, this is how much you need, now what should your protein sources be? A higher quality protein is more digestible and has a better ratio of essential amino acids. In general, animal proteins tend to be higher quality than plant proteins. Their essential amino acid profile tends to be closer to what we need than plants. They're also more digestible, like 5 or 10% different. Should you eat a combination? I'd say yes. Should be a combination of plant and animal. But I wouldn't, there's a lot of people that are anti-animal protein. People make decisions based on a lot of different variables. But for protein quality, there is a difference. One thing you can do, eat more plant protein. Just make sure you mix it based on essential amino acid content. But if we have somebody that's a vegan, I'll add in more protein and try to look for the higher quality plant proteins. But you can get by without the animal proteins. You just it's gotta be more preparation based on that. Okay, questions on that? Thank you guys. I think we're about a minute over, so thank you for that. I'll be seeing y'all soon. If not before, I'll see you Tuesday afternoon.
So should we bring a calculator?
Yeah. Okay.
I have a question about the time of our because I was going to take it in the ERC, but I have another final schedule for 7 to 10.